The genomic landscape of Hodgkin lymphoma, meaning which key drivers shape and affect or underlie the clinical behavior of Hodgkin and its pathogenesis, have been so far investigated mostly by whole-exome or targeted sequencing approaches. So these approaches are absolutely fine and the gold standard to identify the single-point mutations, like mutations of beta-2 microglobulin gene. But they are not really good in defining other features, such as copy number, and in particular, they are not able to define structural variants and mutational signatures, which mutational processes are involved in shaping the cancer genomic and mutational profile. So this is the first study ever where we apply whole genome sequencing to Hodgkin lymphoma cells sorted by patients, both adults and pediatric. We combine this whole genome sequencing with exome sequencing, and what we found has a very dense amount of data showing that there are different drivers as expected on mutations, on copy number, on structural variants like complex event, distinct mutational signatures like mutational process like ApoBec, that is a very important process in hematology and in different solid cancers. But the most striking aspect of our paper is that leveraging whole genome sequencing, we were able to reconstruct the chronological order of events, meaning how these events occur over time. For example, we show that a large chromosomal gain that Hodgkin experience quite recurrently tends to occur very late on time, while in the early phase we have more like mutation on certain driver genes like beta-2 microglobulin or GNA13, or certain deletions like deletion of PTPN1 genes or chromotripsis tends to be very early. The germinal-centered exposure, on the contrary, occur in the middle of the evolution. So we kind of been able to relatively reconstruct the order of drivers. But also, using mathematical and complex genomic investigation, we were able to time in absolute time, like in years, when these chromosomal gains occur in patients' life. So if the chromosomal gain, these large duplications, are late and occur, as we show, at least 6 years before the cancer diagnosis, it means that all the other events that precede these gains occur even earlier. So the Hodgkin pathogenesis, including in pediatric and young adolescent patients, might take over several years. And that's obviously a good window to understand also what happened in these years, how we can eventually prevent, and also localize or understand when the disease was initiated. And that's obviously a very complicated question, but if you know where to look, you may find the answer.